here living in North America, we're pretty accustomed to having food on our table. Most of us can't visualize what it would be like to go weeks upon weeks without eating. But that was the case back in 1944. The German army was losing, so they started hoarding supplies. That led to something known as the Dutch famine. Millions of Dutch citizens were systematically starved of things such as food and water. And among those millions of people were pregnant mothers. Now, a year later, the war ends, the kids are born, and they start growing up. And doctors noticed something weird. As these kids were growing up, they have much higher risk of diseases like obesity and diabetes. And people weren't sure why. But now we know that it was all because of epigenetics, the study of how genes are expressed. It turns out that when these kids were in their mother's wombs, they too were starving. So their bodies did what it does best, and it adapted. All of these kids had a gene called IGF-2, which controls how your caloric intake is stored in your body. And that gene became unmethylated. It became overexpressed. That resulted in a higher percentage of the food that they consumed being stored into fat rather than being used for energy. The body was in a power saver mode. Imagine being so starved that your genes literally changed themselves. And that's probably a big reason why these kids survived. But after they were born and as they grew up, this change left a mark on them. A mark that's gonna stay with them all the way till the day they die. And that's the power of epigenetics. Imagine if we have this tool that could change somebody's life so drastically. What if we could use it to cure one of humanity's worst diseases, cancer? So I started looking into that. But then I quickly realized that there was a difference between cancer and what happened to these kids, in that cancer is much more localized. I needed to find a way to make the treatment way more specific. So I shifted my focus. I started looking into optical epigenetic modification, using light to treat cancer. Now, cancer happens because of uncontrolled cell division. I want you to imagine that your cell is like a car. Is that a Honda? Ew. Okay? The faster that car goes, the faster your cells divide. So let's zoom in on the gas pedal for a second. All the cells in your body have these genes called proto-oncogenes. When they're mutated, they become what are known as oncogenes, the equivalent of pressing down that gas pedal. I started looking into different oncogenes, one known as BRAF, and I chose it to target my treatment. The reason I chose BRAF is for two specific reasons. The first one is that it had impact on the skin, which would make it easy to target with light. Also, it would impact millions of people because BRAF was found to be the cause of the majority of skin cancers. Now, BRAF codes for receptors on the outside of a cell. So imagine that these receptors, they're the Y shape. And these receptors receive molecules, which tells the cells to divide. So when you have more BRAF expression, you have more of these receptors, so there's an increased chance your cell is going to divide. So to do that, to silence the BRAF gene, it would be a process called methylation. Methylation involves adding a chemical called a methyl group to the cytosine base in your DNA. You have your A's, C's, T's, and G's. You add that chemical to the cytosine. So I knew I had to methylate that gene, and I had to come up with a way to do that using optical control. And I came up with a concept that would utilize two different parts, two modules. The first module would be made up of a dCas9 with a molecule called Cry2. Now, a dCas9 is a deactivated version of Cas9. So it's a Cas9 molecule without the scissor parts. So it can target a gene very specifically without actually cutting it. The Cry2 is a type of molecule called a cryptochromatin, which means that it can react to blue light to change its shape. So when the light is active, it can be one shape, but when the light is inactive, it can go to another shape. The second set of molecules, the second module, is going to be made up of a molecule called CIB1 and a molecule called DNMT3A, DNA methyltransferase, which is going to add the methyl groups that we want to silence the BRAF gene. Now, I want you to imagine that CIB1 and Cry2 are like best friends that always want to play with each other. But here's the thing. Cry2's mom says that CIB1 and Cry2 can only play when the sun is shining. That means that only when the blue light is shining on those cells where Cry2 and CIB1 bind to each other. So only when you shine the light will you be able to get the connection between these two modules. And then from there, your DNMT3A can start methylating the target gene. Now, this has advantages over the current methods of cancer treatment we use for cancer because of spatio-temporal control. Spatio meaning space, like location, and temporal meaning time, so the time that we deliver the treatment. Because you're able to control exactly where and when you deliver that treatment. You need the two factors of the light and having that complex on the DNA to deliver your treatment, which is way better than treatments we use currently, like chemotherapy and radiotherapy. There's also potential in the future of changing the Cry2 molecule, looking at different light-reactive proteins from different organisms like algae or plants, and utilizing that so that they're reactive to different types of light, like UV light, which opens up the potential for internal tumor treatment without ever making a single incision. The story of how epigenetics was discovered is a story filled with grief, sorrow, and death. But the way we utilize epigenetics could change that story into the legacy of one of humanity's greatest achievements. Thank you very much.